finding new therapeutic indication for drugs already existing in the market approved by either FDA and or EMA. It's a really faster, cheaper, and safer approach as compared to the traditional developmental processes, drug processes, because through drug repurposing we can bypass many molecular and preclinical steps of a clinical trial. Just to give you a phase one could be skipped because already known safety and pharmacokinetic profiles are available, and there is also a very low risk of failure. Are some very well-known examples like raloxifene, first approved for breast cancer and then repurposed for the treatment of osteoporosis. A very well-known, another very well-known is thalidomide, a sedative withdrawn for a series of dramatic side effects in the 60s, which was repurposed for the treatment of erythema nodosus leprosum, and later on multiple myeloma. Rapamycin in graft versus host disease, repurposed in 1999, and probably the most popular is sildenafil, an antihypertensive drug, which was repurposed by Pfizer for the treatment of erectile dysfunction and marketed as Viagra. Also for drug repurposing, we can ask for the orphan drug designation, the so-called ODD in Europe. There is a similar with EMA, but there is a similar possibility with FDA. And it's intended to promote research on treatments for rare diseases, and White-Sutton syndrome, of course, is a rare disease. In some cases, we can even state that it's an ultra-rare disease, and there is market exclusivity, 10 years protection from similar products, only non-similar products for same indication, free waiver. So through this approach, companies or also academic institutions can in some way get advantages in repurposing already existing drugs. And which is our experience in drug repurposing in the field. Here you can see a picture of White-Sutton syndrome patients. And the rationale of the study is that Perampanel, and the commercial name is Fycompa, is an anti-epileptic drug used in pharmacoresistant seizures, and it acts as a non-competitive antagonist of AMPA receptor. So it plays a role in the excitatory neurotransmission. And why Perampanel in White-Sutton syndrome? Everything started reading a manuscript of Matsumura et al. In 2020, in which mutations in POC-Z cause disruption of neurogenesis through impaired cortical neuronal differentiation and migration, and knockdown mice exhibited an SD-like behavior due to hyperactive excitatory neurons, and Perampanel rescued such phenotype. From this preliminary observation, we decided to design a clinical trial, which is in progress, of course, is still ongoing, in Trieste at the maternal and child health hospital, University of Trieste. The title of the clinical trial is a multicentric drug repositioning self-control case series clinical trial to evaluate the efficacy and safety of Perampanel, improving behavioral symptoms and increasing the quality of life in patients with White-Sutton syndrome, POC-Z related disorders. These are the numbers. These are the numbers of the EU registration, and the main eligibility criteria are the following: molecular and clinical diagnosis of White-Sutton syndrome, males and females aged from 4 to 25 years old, no severe intellectual disability, and no epilepsy. And no epilepsy. So the study design is divided in a series of steps. There is a general assessment, including a complete neuropsychiatric assessment. Then there is the start, the administration of Perampanel at the minimal dosage. Then there are a series of further assessment, either at 6 as well as at 1 year after the beginning of the clinical trial. The clinical trial is designed to last in between 12 and 18 months, if needed, 2 years. And the general assessment includes family and personal medical history, clinical and physical visit, instrumental examinations, and a series of psychological assessment, including a series of scale and tests, cognitive tests, and psychological and psychiatric tests like Vineland, CBCL, and so on. In the beginning, a Perampanel start at 4 milligram per day, and eventually can be increased up to 8, 8 per day. So right now we collected and enrolled in the clinical trial 6 different patients. The first one is a male, 13 years old. The mutation is missense. The phenotype is mild to moderate. The general outcome is extremely positive. The second is a female, 11 years old. At the time of enrollment, the splicing mutation, mild to moderate, and general outcome again is very positive. Then a female, 10 years old, with a fresh mutation, moderate to severe intellectual disability, and the outcome is positive, but not as we were expecting, and nothing compared to the others. There is one male who withdrew after 2 days of the beginning, you being the parents afraid of having this child still in the clinical trial. And then we enrolled 2 more patients, one 7 years old, frame-shift mutation, mild to moderate intellectual disability, and another one male, 6 years old, and nonsense mutation, mild to moderate intellectual disability. And of course, these 2 patients have been enrolled quite recently. Let's present some data regarding the results we got in patient. The first patient is the male, 13 years old. I already described to you. It was showing some mild facial dysmorphism, gastrointestinal involvement, double renal district, intellectual disability, mild, some learning difficulties, emotional disorders, anxieties, issues. He went to, it was enrolled, and for 12 months, he was taking Perampanel 4 milligram per day, and now we have a follow-up of 2 years in this patient. And the outcome is extremely significant in terms of improvements in adaptive levels, improvements in behavior and emotional issues, and in particular, a greater autonomy in personal activity and overall improved quality of daily life, as also reported by parents. I didn't mention you before that both parents, they also have to fill in questionnaires either at the beginning, at 6 months, and at 1 year. Just to give you an example, this boy was so afraid to get out from his home, even to throw away the garbage or going around with the dog. Now he's taking a train alone, flights, he's skiing alone in the Alps, And so it's quality, I mean, his quality of life has improved really significantly. So the second one is a female, 11 years old. She was showing mild facial dysmorphism, hypotonia, overweight, some minor abnormalities at the MRI, yes, at the magnetic resonance and MRI and EEG, mild atrial septal defects. Intellectual disability was mild, ADHD, low frustration tolerance, anxiety issues, and limited social interaction. She completed the clinical trial with the first 2 months, and now she is on follow-up for the 6 months. And also in this case, the improvements were significant in adaptive levels, in behavioral and emotional issues, and also in terms of greater autonomy in personal activities and socialization, as reported by parents. Let me say it in this case, now she has also a boyfriend. Um, the third one, she was presenting some facial dysmorphism, strabismus, hypotonia, overweight, some abnormalities at the MRI and EEG. The intellectual disability was severe, much more severe, mild to severe, motor delay, anxiety issues, and limited social interaction. She was enrolled for 12 months and then considering that the improvements were not that significant, as already included in the study design, it was possible to double the dose from 4 to 8 for an additional 12 months. In this case, there were some, as you can see here, there were some mild improvements in adaptive levels, mild improvement, stabilization of behavior and emotional issue in the second half of the study, parent teachers reports, but not significant switch. In this case, we should consider that this patient was absolutely the most severe one as compared to the three for which we have now completed data. So, in conclusion, drug repurposing in rare disease is a safer, faster, and cheaper approach to add for unmet needs of patients. Our plan is to enroll new Italian patients by the end of 2025 and also during next year. We already visited and prepare for the recruitment another 3 patients, and hopefully they will be enrolled by the end of this year. And the clinical trial, which is an European clinical trial, has been extended to Belgium, Finland, and in the Netherlands. And hopefully also these countries will start recruiting patients very soon. And then finally, what we are working now is to extend the study to another neurodevelopmental disorder, which is CHAMP1 syndrome. The rationale for proposing a clinical trial amendment is that CHAMP1 and POC proteins physically and functionally interact within several molecular pathways, including chromatin remodeling and mitotic progression. Besides indirectly acting on the same pathways by engaging 2 mutual molecules, these 2 proteins also interact directly. Together with HP1 alpha, they form a complex, the CHAMP1 complex, to promote heterochromatin assembly and DNA damage response. CHAMP1 and POC mutation are associated with neurodevelopmental disorders, and CHAMP1 and POC rare disorders share a consistent clinical core, including global developmental delay, intellectual disability, hypotonia, ocular anomalies, and skeletal involvement. And you will hear from Anna Facchini the results of a very accurate work she carried out comparing 2 cohorts of patients, one affected by CHAMP1 disease and the other one by POC, by Sutton syndrome. and in which it's clear that these two disorders share a consistent clinical core. So we prepared the amendment, and of course, we added to the title or CHAMP1 disease. We are now waiting for the final approval in the new CT's platform. To be honest, each time we have to apply for changes in the clinical trial is really a mess, mainly because the local ethical committee in our region is always behaving against clinical trial and new protocols, but this is something we have to deal with. And this is the last slide to thank, of course, Anna Facchini. Most likely you met her last year, and Virginia Musso, who did a very young physician who graduated with a thesis on White-Sutton syndrome last summer, and Domizia Paschetti and all of the patients and the neuropsychiatrics in our hospital, as well as a series of other colleagues who are helping us in recruiting patients, and of course, all the participants to the clinical trial. Thank you so much.
Thank you so very much for presenting your research. This is helpful for us, and we're excited to learn more as your clinical trial progresses.
Yeah, really my pleasure.
Does anybody have any questions for him? Okay, yep, we have a question right here for you.
Okay.
Thank you for the talk. My question is just on, I guess, evaluating the efficacy of this drug and maybe others like it. Like maybe you could talk about what more can be done there. I guess I heard a couple, I know you showed some patients that showed improvement, but I guess I heard mostly qualitative statements about that improvement. And I'm just wondering, like, maybe how you look at that and whether or not the need to, like, get like real tests with objective metrics before and after, things like that. Because I imagine it's difficult sometimes to separate that kind of improvement from maybe what you would see just from getting older, right?
Yeah. These are all metrics. Just to give you an example. For example, this is patient 1. These are all metrics at time 0, 6 months, and 12 months with this scale, which are the Vineland S2. And as you can see, it started, and this is one, the composite score, communication, daily life skill, and socialization. As you can see, it clearly started from an overall score of low, and then this patient reached adequate. And this again, here, if you look at the CBCL from the mother and CBCL from the father, there are a series of improvements, either internalizing problems, externalizing problems, and total problems. And so it's correct. It's not easy to measure behavioral phenotypes, but let me check if I can. But this is carried out through a series of scales like these ones. This is the list of the 7 scales which are currently used to assess time 0 plus improvements at 6 months and 1 year, and then in the follow-up every year, the improvements of our patients, and of course the efficacy of Perampanel. And just to give you an idea, the Vinans are parent report interview assessing adaptive functioning in daily life, covers communication, daily life skill, and socialization. And then CBCL, Parent Report Questionnaire Measuring Emotional and Behavioral Problems, provides score of internalizing anxiety, depression, and externalizing aggression in hyperactive domains. On top of this, what we do is to interview also the teachers at schools to get feedbacks also from them, even if this part um, is not categorized in a specific questionnaire or in a specific scale and score, but this is also considered. In terms of cognitive function, and there were just minor improvements, but to be honest, this is something we are not expecting. Minor improvements, most likely related to the, I mean, to the improvements to the reduction of anxieties. The reduction of anxiety probably helped patients to be more accurate in their assessments, in their scales. Through this approach, there was just minor improvements also in cognitive tests. I don't know if
Got it.
I
Yeah.
was to answer you.
No, thank you. That was extremely helpful. Thank you. So, and the things like VABs too, those are specific neuropsych assessments. Is that right?
Yes, yes,
Okay.
yes, yes, yes,
Awesome. Yeah.
yes.
Thank you.
I didn't mention to you, of course, that during the first month, there is also a pharmacokinetics assessment to measure the dosage through blood specimens to avoid any overdosage. And of course, at the very early beginning, we have all the clinical and instrumental information, including MRI, EEG. But this specific instrumental data are not included in the clinical trial to measure some improvements.
Any other questions? Thank you so very much for your time and for sharing with us today. We look
Okay,
forward to
it was my
hearing
pleasure.
more in the future about this. So thank you very much.
Yeah, I hope next year we will have minimum double of the patients that the one I presented to you today. Minimum,
Yes.
maybe 2 times
Yes.
more.